Okay, thanks for inviting me here. I'm Shujun from Hunan Normal University School of Medicine, China. I'm glad and honored to be here to share the story about immune checkpoint inhibition for triple-negative breast cancer, the current landscape, and our findings that VTCN1 might be a novel therapeutic target. Last year, the incidence of breast cancer worldwide has become the most. Triple-negative breast cancer, the most aggressive subtype, accounts for 10% to 20% of all breast cancers. Due to the lack of estrogen receptor, progesterone receptor, and human epidermal growth factor receptor 2, targeted therapy is limited for TNBC, and chemotherapy is the mainstay of the systemic treatment. As we can see, TNBC has poorer prognosis than other subtypes. They are prone to metastasis and recurrence, so targeted therapy is needed to be developed for a better outcome. Immune checkpoint blockade has showed a promising prospect in a variety of cancers. Here is how it works. Cancer cells could utilize inhibitory immune checkpoint to protect themselves from immune system attacks. Antibodies targeting these immune checkpoint molecules like PD-L1, PD-1, and CTLA-4 could block the interaction and allow T cells to attack the tumors. There are 7 antibodies available now, 2 for PD-1, 3 for PD-L1, and 2 for CTLA-4. As for TNBC treatment, Pembrolizumab, trade name Keytruda, has been approved for high-risk, early-stage TNBC and locally recurrent or metastatic TNBC. Atezolizumab, trade name Tecentriq, has been approved first but withdrawn at last. It's a up and down story, and I'll show more details later. Pembrolizumab is first approved for locally recurrent or metastatic TNBC last year in combination with chemotherapy. The tumors should express PD-L1 with CPS of 10 or more. CPS means combined positive score and is defined as the number of PD-L1 positive cells divided by total number of tumor cells, then multiply 100. This approval is based on Keynote 355 trial. In this trial, PFS was extended by 4.1 months in the Pembrolizumab group. This year, it is approved in combination with chemotherapy for high-risk, early-stage TNBC as neoadjuvant treatment and continued adjuvant treatment after surgery. This approval is based on Keynote 522 trial. In this trial, PCR rate and event-free survival were increased in the Pembrolizumab group. Atezolizumab is first approved based on inhibition 130 trial. In this trial, it was combined with Paclitaxel for advanced or metastatic TNBC whose PD-L1 expression on immune cells was more than 1%. The median PFS was extended by 2.5 months, and the median OS was extended by 7.5 months. But later, in the inhibition 131 trial, the Paclitaxel was replaced by Paclitaxel, and its combination with Atezolizumab did not improve PFS or OS in PD-L1 positive population, so it's controversial for Atezolizumab TNBC indication. This year, Based on inpatient 031 trial, Roche withdraws application for Atezolizumab in early or locally advanced TNBC in Europe. A month later, they withdraw Atezolizumab TNBC indication after talks with FDA. So whether PD-L1 is a good target is questionable. This is a summary of currently known inhibitory immune checkpoints. As mentioned before, PD-L1 might not be a good target since only a portion of TNBC patients express it. As for CTLA-4, its ligands CD80 and CD86 are shelled by the co-stimulatory receptor CD28, which could promote T-cell activation. Other immune checkpoints like LH3, TIGIT, or TIM3 are actively investigated. We are interested in VTCN1 based on our bioinformatic analysis and experiment investigation. I'll share you with the story about how we came up with this molecule. To find the crucial immune checkpoints in TNBC, we first screen the most expressed molecules from a single-cell RNA sequencing data. These authors sequenced more than 1,500 cells from 6 freshly collected, untreated primary TNBC tumors. As the data showed, cells expressing VTCN1 were the most, and the VTCN1 expression were the highest. So VTCN1 might be the main immune checkpoint molecule in TNBC. Next, we compared the VTCN1 expression between TNBC tumors and non-TNBC tumors. We collected 40 TNBC samples and 20 non-TNBC samples and analyzed VTCN1 expression by RHC. Data showed VTCN1 expression was higher in TNBC, so we wonder, could VTCN1 be a therapeutic target? As we know, VTCN1 is a member of B7 family and has 18% sequence homology with PD-L1. Inhibition of VTCN1 could enhance CD8 positive T-cell function and inhibit tumor growth. High expression of VTCN1 is correlated with poor prognosis in several cancers. Our analysis showed among TNBC patients or CD8 positive T-cell enriched breast cancer, VTCN1 high patients had poor survival. This data suggests TNBC cells express high level of VTCN1 to inhibit CD8 positive T-cell function, resulting in poor prognosis by evasion of the surveillance of the immune system, which indicates VTCN1 might be a therapeutic target in TNBC. So next we want to know how it is regulated. As studies showed, metabolites could regulate tumor immunity, like lactate. Increased lactate could promote an immunosuppressive microenvironment by attenuating T-cell activation. To investigate whether metabolites could regulate tumor immunity through VTCN1 in TNBC, we analyzed the metabolism pathways enriched in VTCN1 high TNBC samples. As data showed, 3 glutamate receptor pathways were enriched. Actually, we are excited about this finding because glutamate is like lactate to some extent. First, they are downstream metabolites of glutamate or glucose that are addicted by cancer cells. Second, they are transported upward more in cancer cells. Last but not least, lactate could induce immunosuppressive microenvironment. So we wonder, could glutamate inhibit tumor immunity through VTCN1 upregulation-mediated inhibition of CD8 positive T cells? To answer the question, we first analyze the expression of glutamate metabolism genes. As data showed, the expression of glutamate consumption genes like GS and GLUD were decreased, and the expression of glutamate anabolism gene GLS and glutamate transporter XCT were increased, indicating glutamate might be accumulated and transported outside more in TNBC cells. Next, we measured the glutamate that is transported outside. Cyan columns are long TNBC cell lines. Yellow columns are TNBC cell lines. Obviously, TNBC cells transported more glutamate outside, and this transport is mediated by XCT. So next we want to know, could glutamate regulate VTCN1 expression? By adding exogenous glutamate to the media, we found VTCN1 expression was increased, and when XCT was interfered, VTCN1 expression was decreased. Among TNBC samples, VTCN1 expression was positively correlated with XCT analyzed by RHC. Now we have known that more glutamate is transported outside in TNBC cells and upregulates VTCN1 expression, but how the signal is transduced into the cell. After some paperwork and analysis, we have came up with this regulation pathway. First, glutamate binds to its receptor, which could activate ERK. And we also screened the c-Fos as the transcription factor of VTCN1, which could be activated by ERK. So, we hypothesized that VTCN1 could be regulated by glutamate receptor ERK c-Fos pathway. Glutamate has 8 metabotropic glutamate receptors, so we first analyzed the expression using TCGA data and found glutamate receptor 6 was higher in TNBC samples, and the field change was the largest compared with non-TNBC samples. While among TNBC samples, VTCN1 expression was positively correlated with glutamate receptor 6, and when the receptor was overexpressed, VTCN1 expression was increased. Now we have got glutamate and receptor 6 could induce VTCN1 expression. Next, we interfered XCT and found the phosphorylation of ERK was decreased. When we overexpress this receptor 6, ERK phosphorylation was increased, and when c-Fos was overexpressed, VTCN1 expression is also increased. So we have confirmed this part, and the regulation of c-Fos by ERK is still missing. We are working on it. In summary, we found VTCN1 is higher in TNBC and predicts poor prognosis. Glutamate efflux is increased and induced VTCN1 expression by metatropic glutamate receptor 6, ERK, c-Fos pathway in TNBC. However, a lot of work remains to be done, like the inhibition of CD8 positive T cells by glutamate. Anyway, our work reveals VTCN1 might be a novel therapeutic target for immune checkpoint blockade. It's our lab. This is Professor Deng Ximing. He gives me a lot of support, and it's me. This work is done by this student and is funded by National Natural Science Foundation of China. Okay, that's it. Thanks for your attention.